Hi, my name is Megan Ohol, and I'm a research intern in the Beeler Lab. Cancer is one of the world's deadliest diseases and a leading cause of death in the United States. In certain cancers, the overexpression of proteins plays a large role in uncontrolled cell division, which is essentially what cancer is. Protein overexpression occurs when too many copies of a protein are created, which becomes problematic when these proteins are subsequently not regulated properly. At first, small molecule inhibitors were created to try and inhibit the function of these overexpressed proteins. However, there has been a recent uptick in proteins developing drug resistance to the inhibitors, so the inhibiting function doesn't actually work. And this is where my research comes into play. My research focuses specifically on a protein-to-protein interaction in breast cancer. A transcription factor called FOXP3 is highly overexpressed in breast cancer patients. FOXP3 is responsible for regulating the cellular activities of regulatory T cells, which are an important part of the body's immune system. The high activity levels of FOXP3 are specifically related to another protein called TIP60, which enhances or amplifies the FOXP3 protein. TIP60 is like a speaker, for example. Think of giving a speech to a large crowd. Without any kind of amplifier or microphone, you can only reach a few people. But if you have something there, then you can reach the whole crowd. And that's essentially the role of TIP60. My research project has been to create a protein degrader called a PROTAC that targets TIP60 in order to stop the overexpression of FOXP3. A PROTAC, or a proteolysis targeting chimera, is a molecule made up of an E3 ligase binding domain and a target protein binding domain attached by a linker. Think of two magnets attached by a string. One of the magnets is going to attract the E3 ligase, and the other one is going to attract the target protein, and then the linker is just the string that holds them together. The role of the E3 ligase in the PROTAC is to attach a molecule called ubiquitin to the target protein. This ubiquitin molecule tags the target protein for degradation, and a protein complex that's part of the cell's waste removal system called a proteasome will find and destroy the target protein. My PROTAC was successfully synthesized through a series of reactions by attaching an E3 ligase binding domain and a TIP60 binding domain with an amine linker. The end product of my experiment could potentially become a new cancer therapy that greatly reduces the problem of drug resistance. The protein can't develop any drug resistance because it's completely degraded and it's gone. There's no way for it to have the time to form any of that resistance because it's destroyed. The resulting product will be sent to collaborators for further biological testing to see what effects the PROTACs will have on living cells. Overall, this PROTAC has the potential to save thousands of lives effectively, overcoming some of the major obstacles that today's cancer therapies currently face.